Assalamualaikum everyone, welcome back to this channel. So this is me Alia Wahid, and in this video we are going to start a new chapter of microbiology, and this chapter is about the disinfection. So in the previous lecture we have discussed about the sterilization, we have discussed all the types of sterilization, physical method, chemical method, and we have discussed about how to sterilize with the help of heat and with the help of radiation. So in this lecture we are going to start the second method of killing the microorganisms, and this method is called as disinfection. So actually disinfection is a process of cleaning the microbes from the non-living objects. So this is called a disinfectation. So what is a disinfectant? So it is the antimicrobial agents that are applied on the surface of non-living objects to destroy the microorganisms. They are only apply on the surface of non-living things. They cannot be applied over the surface of living organisms. So here I have written about the antibiotics and antiseptics. It is just for additional information for all of you. So antibiotics are going to destroy the microorganisms which are present within your body, not outside on the surface of skin. But the antiseptics are used to destroy the microorganisms which are on the surface of the living things like your skin. So all the bacteria, fungi, microbes which are present on your skin, they are killed by antiseptics. And the microbes which are inside your body, they are killed by antibiotics, which are the antibacterial agents. Now they have given some methods of disinfection. Two methods, physical and chemical method. Let's discuss the physical method first. Physical method is again divided into the two methods, like first is the direct boiling and second is the pasteurization. Now let's discuss both of these methods. So first is the direct boiling. So we are going to boil the non-living objects, all the non-living objects, like if you want to remove all the microbes from the surgical instrument, what you will do, you will boil them in the water. It will kill all the microorganisms present on it. So example, like water, after the boiling it act as a disinfectant, so it will kill all the organisms or microbes over the non-living objects. Second is the pasteurization. So pasteurization is a process of heating a food or a liquid into a specific temperature. So you will set it at a specific temperature, or then you will heat your food or your liquid. Food or liquid will contain like foods which are prepared in the industries. They undergo the process of pasteurization in order to kill the microorganisms on them. And some liquids which are prepared in the industry, like milk is being packed, like the Olper company is going to pack it. They are heating that milk at a specific temperature for a definite time, okay, and then it will immediately cool. So by cooling it, it will destroy all the microorganisms present on it. It has some advantages, like it will stop the microbial growth. So in our milk, in our food, a lot of microorganisms are present, and many of microorganisms which are present on them, they are pathogenic in nature. They can cause various diseases to you, so therefore we should stop them. So this process of pasteurization will slow their growth and also decrease the number of these pathogenic bacteria which are responsible for causing you a lot of diseases. Now we are going to discuss the method of pasteurization in the next. So now we are going to start the method of pasteurization. So we have three method of pasteurization. So the first method is the long time and low temperature method. So long time mean it is kept at the long time, mean about 30 minutes, and the temperature is low at that time, and the temperature is 63 degrees centigrade. By keeping at this temperature and pressure, you will keep your food or any liquid in it, and it will kill all the microorganisms. Second method is the short time and high temperature method. So in this method, you will keep at the temperature of 71.7, and the time is 15 to 20 seconds. And after that, the third method is very short time and ultra high temperature method. So in this method, you will keep very short temperature, but for very short time, mean about 3 to 1 seconds only, but the temperature is very high for that time, and it becomes 140 to 150. So these are the three methods of pasteurization. What are the aims of pasteurization? So it is written here. So it is going to prevent all the milk-borne diseases, and it kill all the pathogenic bacteria which are present in your food or liquid. Now we have discussed all the physical methods of of killing the microbes. Now we are going to start the chemical disinfection, mean we are now going to use chemical agents to kill all the microorganisms. So what are the chemical agents which are used? So here I have written some agents like alcohol, hydrogen peroxide, aldehyde, acid and bases, heavy metals and detergents. They are used for that purpose. Now we will discuss all of these agents one by one. So the first is alcohol. So alcohol is very popular antimicrobial agent. What is its mechanism of action? So it is causing the protein denaturation in microbes. So you know, every living thing is made up of protein. So microbes like bacteria, fungi, they also contain proteins in them. So if the protein denaturations occur, then the microbe cannot survive, they will die. So alcohol is causing the protein denaturation inside the microbes. So examples of alcohol is 2-methyl alcohol and ethyl alcohol. Methyl alcohol is less effective, but ethyl alcohol is 50 to 90% effective. So therefore, it is mostly used for that purpose. It is also non-irritating, cause no irritation to the food or the liquid, and it's also inexpensive. So therefore, it is mostly used. Second is the hydrogen peroxide. So hydrogen peroxide formula is H2O2. So its mechanism of action is that it will release a free radical, which is the hydroxyl free radical. This hydroxyl free radical have a ability to react with the oxygen radical, and you know that the oxygen radical is mostly responsible for causing the cancer like diseases. So hydrogen peroxide here is acting as an antioxidant agent. So it is used for the sterilization of the contact lenses. So you know it, on the contact lenses lot of bacteria's population can occur, and when you wear these contact lenses it will cause severe kind of infections in your eye. So therefore the solution of hydrogen peroxide will use to kill all the microbes which are present on the contact lens as well as on the surgical instrument. So now we have discussed the first two chemical agents. Now we move toward the third chemical agent which is used for that purpose. So here I have given the third agent, which is the aldehyde. So in the aldehyde, formaldehyde is a gaseous form which is used. So mechanism of action is that it will react with the nitrogenous compound. And what are the nitrogenous compound? They are the proteins. So proteins contain nitrogens in them, so therefore they are called nitrogen containing compounds. So they will react with them and they will denature them. They will destroy the proteins of the microbes. So let's discuss the fourth chemical agent, which is the base and acids. So various kind of bases and acids are used for that purpose. Their mechanism is that they increase and decrease the pH. So by increasing the pH, some bacteria cannot survive the increase in the pH, they will die, and some bacteria cannot survive the decrease in the pH, so they will also die. So the acids and base are added, so do change the pH. So bases are used as a cleaner, as a detergent, but acid is used for the food preservation. Some heavy metals are also used for that purpose. They will also bind to the nitrogenous compound of like protein. Heavy metals examples are mercury, silver, and gold. For example, organic mercurals are present. They are added in the cosmetics. So actually, they are killing the bacteria. Now let's discuss about the detergent. So detergents, they are the organic molecule. They serve as a wetting agent. So examples of them are the sodium lauryl sulfate. Mechanism is that they are going to denature of protein. Uses in the hospital floor, washing the dishes and utensils. So this was all about the today's lecture. Meet you soon in the next lecture. Allah Hafiz.